Hi, we're PharmaCure, and we're here to change the fate of the future. With your support and funding, we aim to develop novel anti-malarial drugs that aim to combat the issues that the world is currently facing with regards to anti-malarial drug resistance.
We hope to create more effective antimalarial drugs through enhancing binding affinity and improving potency of our compounds. In doing so, we also hope to maintain good pharmacokinetics and reduce side effects of current drugs.
As there is a clinical need for a new medication in this area, we believe our compounds are fundamental. Therefore, we hope you will consider our proposal of the potentially life-saving compounds we have created.
Malaria occurs in more than 90 countries worldwide. On average, it kills one child every minute and 500,000 people every year. Malaria is not only preventable but also curable, yet around 3.4 billion people are at risk of malaria. Malaria is more prominent in tropical and subtropical areas of the world. People living in the poorest country are most vulnerable to malaria, along with people who lack or have little immunity against the disease. The economic cost of malaria in Africa is estimated to cost more than $12 billion per year, and is thought to cost households around 32% of their entire monthly income. Malaria incidence and mortality rate fell by 37% and 60% globally. However, there are still 214 million people suffering from malaria and 438,000 estimated deaths due to malaria. African region accounted for most global cases and deaths of malaria, followed by Southeast Asia and Eastern Mediterranean region. These striking figures highlight the need for an increase in efforts to reduce the burden of malaria in many places. There is also a concern that P. falciparum in Greater Mekong subregion has become increasingly resistant to anti-malarial medicine, including artemisinin-based combination therapy. There is a real risk that multi-drug resistance will soon emerge in other parts of subregion, as well as the spread could pose a major public health challenge and jeopardize important recent gains in malaria control. Not to fret, together we can find a solution to this worldwide problem.
Amodiaquine is chosen as lead compound as it shows exceptional efficacy towards chloroquine-sensitive strains K1 and chloroquine-resistant strains HP3 of Plasmodium falciparum. The introduction of phenyllinker increased its activity against chloroquine-resistant parasites. It also consists of planar 4-aminoquinoline ring, which engages in pi-pi stacking interactions with the heme protoporphyrin ring. The protonation of the nitrogen allows the drug to accumulate in the parasite's food wall. Lastly, there is potential for intramolecular hydrogen bonding between the protonated amine and hydroxyl group due to the proximity between the two. However, amodiaquine was withdrawn as a licensed drug due to its hepatotoxicity side effect, which is caused by the production of toxic quinoimine. In addition, amodiaquine has low bioavailability due to extensive dealkylation of diethyl amino group. Amodiaquine also has low molecular weight to provide room for improvement and make structural modification to make new anti-malaria drug which could save the life of millions.
Hi, my name is Amira. I'm the medicinal chemist. For this project, we propose to make 20 compounds with different modifications, which we hope to show improvement in both activity against chloroquine-resistant clone and combating toxicity effects in comparison to amodiaquine. The OH group in amodiaquine made the compound toxic as it causes the production of reactive metabolites. Therefore, we interchange the OH and the diethyl amino side chain as it will reduce toxicity by preventing the production of toxic metabolites. We also replace the OH with fluorine, as literature suggests that substitution of this group reduces toxicity as toxic metabolite will not be produced while still retaining substantial antimalarial activity against chloroquine-resistant parasites. Besides, it was found that the diethyl amino group is susceptible to metabolism. Therefore, we replace it with piperidinium, pyrrolidinium, N-butyl, and morpholinium group, which are more stable and will lead to substantial increase in antimalarial activity. In addition, we also modify some of the compound by oxidizing the nitrogen in quinoline ring, as literature suggests that it will result in activity of 3 times higher than amidoquine, which may be due to increased binding affinity at the target site. It was also believed to increase the activity of compounds in vivo. And the last modification was the addition of methoxypyridine group to the quinoline ring. This is because literature suggests that compounds that contain this group shows higher activity against chloroquine-resistant parasites.
Hi, I'm Lauren, our team synthetic chemist. To synthesize our compounds, we first require our 5R1 amine groups: diethylamine, morpholine, N-tertiary butyl, piperidine, and prolidinol. We react these with M-hydroxyacetanilide under the Mannich reaction, using the conditions of formaldehyde, ethanol, and heat to make our intermediate. This is then hydrolyzed using hydrochloric acid and water to make our second intermediate. This then undergoes a variety of nucleophilic substitutions with heat and solvent to make our final compounds. Firstly, with 4,7-dichloroquinoline to make our first set of compounds. Secondly, with the N-oxidized version of 4,7-dichloroquinoline to make our second set of compounds. And thirdly, with 7,10-dichloro-2-methoxybenzo-1,5-naphridine to make our third set of compounds. To make our fourth set of compounds, we need to use the carbonylation reaction. We react our amines with 2-bromo-1-fluoro-4-nitrobenzene with the conditions of carbon monoxide, palladium 0, ethanol, and hydrochloric acid to make our intermediate. This then undergoes reduction reactions. Firstly, with iron acetic anhydride acetic acid in 60 degrees, and this intermediate is then reduced again with lithium aluminium hydride and diethyl ether to make another intermediate. This intermediate undergoes nucleophilic substitution with 4,7-dichloroquinoline under solvent and heat to make our fourth set of compounds.
Hi, I am Pei Yan. I am the medicinal biologist. Our compound could be susceptible to a variety of metabolic pathways in vivo. For example, the N-oxide can undergo reduction in the liver by N-oxide reductase, and the oxygen will be cleaved off. The methoxy group on the acyl-N-cridine ring may be susceptible to O-demethylation, forming alcohol group on the acyl-N-cridine ring. Phenol is metabolized via O-glucuronidation or sulfation during phase 2 conjugation metabolism. Morpholine ring could be degraded by enzymatic or non-enzymatic oxidation, followed by ring opening step to form carboxylic acid metabolite. The pyridine ring and piperidine ring are subject to hydroxylation and oxidation, followed by ring opening step to respective carboxylic acid metabolite. The terpretalamine has branch alkyl group which make it stable against the alkylation. However, it may be susceptible to hydroxylation and oxidation. The diethyl and amino moiety undergo N-deethylation to form mono-dis-ethyl metabolite, which can be further metabolized via second deethylation to form inactive bis-dis-ethyl metabolite. The fluorine is quite stable against metabolic attack.
Hi, my name is Priyanka and I'm our team's medicinal biologist. Initially, we intend to use in vitro bioassay methods, especially the hyperxanthine incorporation assay, to screen all 20 of our compounds. This is because this method is cheap, rapid, and high throughput. Later, we plan to take forward the best compounds and screen them using in vivo assay methods. This will allow us to evaluate the compound against rodent strains of malaria parasites. This process also allows us to maintain low costs and sustain ethics without compromising comprehensive evaluation of our compounds. The best compounds chosen will be those that exhibit an IC50 of less than 20 and those that exhibit exceptional efficacy against strains resistant to chloroquine. It would be ideal if the compound also exhibited efficacy towards chloroquine-sensitive strains too. We also intend to test the N-oxidized compounds to see its effects in an in vivo system. We have predicted the physicochemical properties of our compounds using chemical eyes. This is to make sure that our compounds meet the necessary Lipinski requirements, which allow our compounds to be considered as orally bioavailable. However, We intend to carry out further essays to analyze the metabolic pathway of our compounds to further determine the bioavailability of our drugs. This will allow us to make any modifications to our drug compounds where necessary.
We hope that you can see the importance of compounds that we wish to synthesize. With your support and funding, we aim to combat the issue the world is currently facing against anti-malaria drug resistance.
Our team of expert medicinal chemists and biologists require 5-month fundings to synthesize and analyze our 20 proposed compounds, 1 month for SAR studies, 1 month for compound synthesis, and 11 weeks for analyze studies to confirm our drug effectiveness.
We believe the high-quality compounds that we have developed pose an unmissable opportunity to tackle the burden that is malaria, and we hope that you can help us change the future for the better.